This video is a continuation of the previous video on genetics. We're going to start by looking at gene mutations. A mutation is a sudden change in the genetic makeup of an organism, so in the DNA of an organism. A gene mutation results from a change in the sequence of nucleotides in a DNA molecule. So we know that in the nucleotides there are 4 bases, adenine, thymine, cytosine, and guanine in DNA. And if there is a change in the sequence of these nucleotides, so the sequence of the nitrogenous bases, then this is what we call a gene mutation. A gene mutation causes a change in the information that the genes give to the cell, i.e. the codons will be altered because there's different sequence of nucleotides, which means that different anticodons will be coded for and therefore different amino acids will be brought. And because of this, there will be a faulty protein being made, or not, or no protein at all, or a shortened protein, or it could be the same protein if we look at the redundancy of the code, but we're not going to focus on that. For now, we just know that a gene mutation causes a change in the information a gene gives to the cell, and therefore a faulty protein is made. A mutagen is any factor that increases the rate of the mutations. So some mutagens could be things like ionizing radiation, mutagenic chemicals, viruses, and microorganisms. How do mutations affect organisms? Well, they can have a variety of different effects on the organisms depending on the type of the mutation. So it could be a chromosomal mutation, a point mutation, somatic mutation, gametic mutation, a substitution mutation, an addition mutation. These are all different types of mutations, and the names vary in the detail of the mutation, but for now, we're not going to worry too much about those. They can also vary, their effects can vary depending on the importance of the piece of genetic material that's affected and whether the affected cells are germ cells, so gametes, or somatic cells. Sorry, excuse my dog barking in the background. Germ cells are often more, have a bigger effect on the organism because it can be passed on to future generations, and every cell develops from that cell. So often all the cells will have that mutation. So somatic mutations occur in body cells. They aren't transmitted to the next generation. Often somatic mutations can cause cells to become malignant, resulting in cancer. In plants, they can be transmitted to the next generation by vegetative propagation. Gametic mutations occur in the reproductive organs, so the ovaries, testes, anthers, or embryo sacs, and they produce changes to the genes within the gametes. These mutations are also called germline mutations, and they can lead to variation in the offspring. Eventually, by accumulating these changes, a new species may evolve, which is a process called speciation, and this plays a very important role in evolution. So as more and more mutations happen and these changes become more common within the population, so more offspring are exposed to these changes, a completely new species may evolve from the ancestral organisms because the offspring have so many new different mutations, and this is speciation. Gametic mutations can also lead to hereditary diseases, which are harmful. In the mutations that cause evolution, these are usually beneficial mutations because they allow the population to survive, whereas diseases that are passed on are, of course, usually harmful. Some mutations are neutral, which means that they either don't actually affect the the phenotype of the organism, or they do affect the phenotype, but it has little to no actual consequence. For example, tongue rolling. You can see that it does affect the phenotype if you're able to roll your tongue, but it doesn't affect your life. You're not going to be more or less adapted or something like that. It's not going to affect whether you have a greater rate of survival. So neutral mutations are also known as harmless or silent mutation. Beneficial mutations are mutations that result in a change in the phenotype and allow the organisms to be better adapted to new environments and allow them to breed more successfully. This is what natural selection is. It relies on beneficial mutations to select the better adapted organisms to survive and breed. In time, the whole population will have the new genotype, so we'll have the mutation, and a whole new species will have formed. So speciation has occurred. Some examples of beneficial mutation could be the antibiotic resistance in bacteria, this is especially problematic because bacteria reproduces so fast and viruses reproduce so fast. So the mutation can spread very quickly to the entire population. And this is why also beneficial mutations do occur more often among viruses and bacteria. There's also a peppered moth, which had a beneficial mutation during the Industrial Revolution, where it used to have a pale form, but it mutated so that there is a dark form in some parts of Great Britain, which allowed it to be better camouflaged within the coal dust. So they were less likely to be eaten by predators. So it was actually a beneficial mutation. Harmful mutations are the more common ones that we see in animals and in humans, and they cause genetic disorders. Humans have over 100 syndromes caused by chromosomal abnormalities, such as Down syndrome, and more than 6,000 diseases caused by the inheritance of mutations in single genes. So these would be point mutations. Things like cystic fibrosis, sickle cell anemia, Tay-Sachs disease, Huntington's disease, and color blindness. These diseases are usually inherited as autosomal recessive traits. So autosomal refers to chromosomes that aren't the sex chromosomes. So these would be non-sex linked traits. But as we looked at in the previous video, something like color blindness is in fact a sex-linked trait. Heterozygous parents will each have one normal copy of the gene and one mutated non-functional copy. And because these diseases are usually recessive traits, the parents wouldn't show the symptoms, but they'd be carriers. Homozygous recessive individuals actually exhibit the disorder. They show the symptoms because both gene products, so the proteins made by both alleles, are missing. They have two recessive alleles. This is one reason that marriage between close relatives is discouraged, because two genetically similar adults are more likely to pass on two copies of a defective gene to their child. So to have a child that is homozygous recessive for a harmful trait. Something like albinism is a harmful mutation. It's a genetic disorder caused by a recessive allele that lacks the instructions for the production of the pigment melanin. So melanin is the pigment that gives the color to skin, hair, and eyes. And a person with albinism would lack this pigment melanin because the recessive allele doesn't have the instructions to produce this pigment. Albinism isn't sex-linked. It occurs equally in males and females. The skin is very pale due to the shortage of melanin, and hair is blonde and eyes are blue. Eyes can even appear a little bit pinkish. People with albinism are extremely sensitive to light. They often have weak vision, and they're at high risk for skin cancer because their skin is so light. They obviously are homozygous recessive because they carry the double recessive alleles, and that's why albinism is expressed. If a person is heterozygous, they'll have the normal skin color, but they are a carrier of the recessive gene, so they can pass it on to their children. Sickle cell anemia is another harmful mutation. It's a point mutation, so only one nucleotide is actually affected. And it's caused by a gene mutation, obviously. It occurs mostly in the malaria areas of Central and West Africa, as well as in the Mediterranean. In the gene that codes for the formation of hemoglobin, so a protein on red blood cells, the nitrogenous base adenine is replaced by thymine, so that's your point mutation. It happens during DNA replication. It leads to the formation of abnormal hemoglobin, known as hemoglobin S, so they'll be sickle cell shaped, the red blood cells, instead of the normal biconcave discs. Because they're sickle shaped, they can't effectively fulfill their function of transporting oxygen, which is a very important symptom of sickle cell anemia. Some of the sickle cells shaped red blood cells block the capillaries, and they prevent the movement of the blood cells that are actually normal shaped. This leads to poor circulation and anemia because there's not enough oxygen on the sickle cells. And then some of the sickle cells can block the capillaries, stop the normal red blood cells from getting through, which can cause something like a stroke or a heart attack, etc. And because of this, people with this disease have a very short life expectancy. One of the it's a side effect of sickle cell anemia, but it's actually surprisingly a beneficial side effect, is that people with sickle cell anemia are actually resistant to malaria, which is one of the reasons why sickle cell anemia occurs to the people in the malaria areas of Central and West Africa. Because people with sickle cell anemia, because they can't get malaria, even though they have a low life expectancy because of this mutation, they may still have a higher chance of reaching adulthood and the age of sexual maturity because they aren't exposed to malaria. So they can have children and pass on this disease, which is why it's more prevalent in these areas. A single nucleotide polymorphism, also called a SNP, is a genetic variation in a DNA sequence that occurs when a single nucleotide is substituted for another nucleotide. So we know that that's called a point mutation. But SNPs are specific types of point mutation. They are point mutations that are considered to be evolutionarily successful. So they have become common enough that they can occur in a significant proportion of the population of a species. So they won't just occur in like 1 or 2 or 3 individuals, they'll occur in a large number of individuals within the population. They occur throughout the human genome, about 1 in 300 nucleotide base pairs, and there are about 10 million SNPs within the 3 billion nucleotide human genome. Modern insights into these genetic variations are changing the understanding of inheritance. So most commonly, these variations are found in the non-coding DNA between genes, simply because the coding DNA for humans is often very similar. There often aren't that many point mutations because most proteins coded for in humans are the same throughout all humans. Things like proteins coded for the blood cells, for eye color, for hair color, for etc. Those are everybody has those proteins. These SNPs can act as biological markers which help scientists locate genes that are associated with disease. And they also show different population groups from different areas, because often population groups will have the same SNPs, which means then you can see that people have occurred in the same area. For example, like Northern Europe, Southern Africa, Western Australia, whatever. People from those areas will have the same SNPs. So if we then move on to look at a genome, the genome is the complete set of genetic instructions necessary to create an organism. Almost every somatic cell in the body has a complete copy of the genome, and each species has a unique genome, although the genome of individuals of the same species may vary slightly due to mutations. Usually within the species, it has exactly the same genome, that's the general rule, but if we look closely, we can see that there might be a few differences within individuals of the species because of mutations. So the Human Genome Project was an international scientific research project set up in 1990 with the primary goal of determining the sequence of chemical base pairs which makes up the human DNA. So sequencing the human DNA. They wanted to identify and map all the genes of the human genome from both a physical and a functional point of view. There's often questions asked about this in exam papers, just simple ones such as what is the Human Genome Project, what's the primary function of it, what are some of the advantages of this, what can it find out, and that could be things like it could find out genetic disorders, which nucleotides affect the disorders, we could then look at research into fixing them, etc. Just as an interesting fact, humans only have 1% of unique genes, so 1% of genes that we don't share with other species. What's genetic counselling? So genetic counselling provides information and support to families who have members with birth defects or genetic disorders like Down syndrome, Huntington's disease, and so on. As the hereditary conditions may be passed on from parent to children, many genetic diseases or disorders tend to run in families because they passed on from parent to child and from parent to child. Genetic counselors then study the family and the medical histories, and they analyze inheritance patterns and the risks of the problem recurring. So they look at the histories, the medical histories, the patterns of the past, and they see, okay, obviously they can't be exact, but they can say, okay, this is the probability that you will have children with this disease, or that your children will have children with this disease, and so on. It aims to provide families with the options for the best ways to manage the disorder, and they can refer families or individuals to appropriate social support structures. So some sort of issues that can be discussed is if this couple comes to the genetic counselor and they say, okay, we've got a history of this disease in the family, I know that I'm a carrier and my husband's a carrier, should we have children? And the genetic counselors can say, okay, well, these are the symptoms of the disease, this is the life expectancy, this is the probability of you having children with the disease. and then they can discuss, should we have children or should we adopt a child? Genetic counselors can also say things like, is artificial insemination by a donor or embryo donation a good alternative? Is in vitro fertilization and the testing of embryos before they're implanted a good option? And then something that's also added in the end is, should cousins contemplate having children? This is quite a stupid addition, but it's basically saying, is incest a good thing? And obviously the answer is no, because as we said, among others, I can't remember where we said it, but we said that because families often have the same genes or similar recessive disorders, and they pass on across families, incest isn't a good thing because the recessive gene is more likely to show up. So what is a family tree? You'll see this sort of thing, which is called a family tree. You'll see it very, very often. It's almost always asked in a past paper for a few marks. So in humans, the pattern of inheritance of a particular characteristic can be investigated by researching a family tree or a family pedigree. They show the genotypes and the phenotypes of several generations of individuals in a family. Individuals could have a normal phenotype, but when we look at the genotype, they actually are a carrier of the genetic disorder, where the mutant allele is recessive. So you can look at this family tree and see whether the parent was a carrier if their child has the disease. And I'll look at it in a bit more detail just now. So a family tree can be constructed to predict whether a couple is likely to pass on a genetic disorder. You can get a family tree for sex-linked and for autosomal disorders, and they'll usually tell you whether this disorder is autosomal or sex-linked. But sometimes you might have to look and see, like, if only the males are showing the disease, it's usually likely to be a sex-linked disease. Okay, so that's the this would be a family tree. If we look at this one, the important thing to know is that females are circles and males are squares. They'll usually give you a key, but if they don't, it's good to know. The colored in dots, they'll usually do it in black and white, where the black colors are the ones who are affected, so the people who show the symptoms of the disease, and the white colors are the ones who don't show the symptoms. So if they're white, they might be a carrier, but they don't show symptoms in their phenotype. I'm just going to look at this diagram because it's a bit bigger. So in this case, red is affected and blue is they're not affected. So if we just start here at the top, these two, the original parents, a female is affected, the male's unaffected. Immediately we can see that this is probably not a sex-linked disease because the female has it. Obviously we know that some females can get some sex-linked diseases, but chances are this would be an autosomal disease. Now we can see that this parent, or these pair of parents, had 1, 2, 3, 4, 5 children. These over here aren't children because they don't have lines connecting them. They had 1, 2, 3, 4, 5 children. They had 3 males, 2 daughters. Both their daughters were affected by the disease. 1 son was affected and 2 sons were unaffected. Immediately we can see then that we don't know if this is a dominant or recessive disorder. But if let's assume it's a dominant disorder, that would mean that the male is homozygous recessive. Okay, sorry. Let's assume it's a recessive disorder because that's more common, the question. Let's assume it's a recessive disorder and we know it's not sex-linked. This would mean that the female is homozygous recessive for the disorder and the male is either heterozygous, so a carrier, or homozygous dominant. We don't know. The question is, for example, let's figure out what this male is. Is he heterozygous? Is he homozygous dominant? And we need to look at the children to figure that out. So we know that the daughter, this daughter must be homozygous recessive because we know it's a recessive disorder. This daughter is also homozygous recessive. That means that she must have received 1 allele from her mother, the recessive allele. From her father, she needs to have also received 1 allele, right? Because otherwise she can't be homozygous recessive. That means that her father must have had a recessive allele in his genotype, which means that he must be heterozygous. He can't be homozygous dominant. And we know that because we've spoken about before Mendel's law of dominance, where a homozygous recessive and a homozygous dominant individual, when they mate, they'll only produce heterozygous organisms, heterozygous offspring. And we can see here that the offspring aren't heterozygous clearly because some are showing the disease and some aren't. So that's how you'd start to look at a family tree and to use it to work out individuals' genotypes. They may ask you sometimes to work out, is this disease recessive or dominant? Or they might ask you, what is the genotype of this individual? Or if these two individuals had an offspring, what is the likelihood of them of their offspring having the disease, etc. The easiest way to do it is look at what you know. So, for example, if we knew here that this disease was recessive, we know without a doubt that this individual is homozygous recessive, as this individual, this one, this one, this one, this one, this one, all the affected ones. So you write out their genotype. Let's say we use the letter H. We'd write out H, lowercase h, next to all the affected ones. We've now used it to figure out that this one's heterozygous. So capital H, lowercase h. We now see here, we look at these individuals. We know that this is homozygous recessive. We don't know what this person is, but we can see here that they've produced at least 2 offspring that have the disease, which means that this person has to also be heterozygous. And that's how you'd look through the tree, writing down next to each individual what you think their genotype is. That's the simplest way to solve trees, and just practice with family trees over and over. At first, they can be a little bit confusing because you feel like you have no information. But as you look, you can make assumptions and then look at the parents and say, okay, if I've assumed that the son is homozygous dominant, what does that say about the parent? And if you can see, well, obviously the parent can't be like that, then you know that your initial assumption was wrong, and so on. If you do have questions about family trees, you're welcome to send me a message and I'll answer them for you. So then if we move on to look at genetic engineering. Genetic engineering is the direct manipulation of the genes in an organism to obtain a desired characteristics, and the relevant genes from a cell in one organism is transferred to a cell in another organism. The organism that receives this gene, so the organism that has been genetically modified, genetically engineered, is called a transgenic organism. An organism that is genetically modified, we say, is GM, or genetically modified organism is called a GMO. In an exam, you can use the letters GM or GMO, just specify first that this is what they stand for. We already know that a gene is a segment of DNA strand, and this segment provides the code for the synthesis of a particular protein. Different genes code for different proteins, and some proteins can be things like enzymes, building materials, hormones, immune response, and reserve fuel, so reserve energy. Genetic engineering can also replace faulty or missing genes that cause disorders or diseases. This is what we call gene therapy. It ensures that the correct protein is synthesized, and the basis of this process includes the following steps. So first, geneticists or the people performing this process will identify the relevant gene in a healthy cell. They'll extract it and they'll cut it from the DNA. They use restriction enzymes to cut it from the DNA. This isolated gene, so this gene that they've just extracted and cut from the DNA, is then inserted into the defective cells, so the faulty cells, by means of vectors, so viruses or bacteria. And a vector is simply an organism that transferred something. In this case, an isolated gene is transferred by a vector to the defective cell. This isolated gene then becomes integrated in the organism's genome, and because of this, the correct protein is synthesized and the desired trait is expressed in the phenotype. Genetic engineering results in the formation of a new DNA sequence in the recipient cell. So the nucleotides or the nitrogenous bases have been replaced by one or more new nitrogenous bases. And the new DNA that is formed is known as the recombinant DNA. The word recombinant refers to a new combination of DNA in the organism because of this new gene that is being received. The organism that possesses the recombinant DNA is called the transgenic organism, and it can now synthesize the protein coded by the new genes. The integration of genes for therapeutic purposes in cells with faulty or missing genes is called gene therapy, which I mentioned above. So this is the process of genetic engineering. You find the organism with the desired gene, so for example, the red pigment gene. You extract the DNA from that organism. You locate and copy this desired gene from thousands of genes that were extracted. This is called gene cloning. So when you extract the DNA, you extract the whole genome, and you only want the specific gene. So you use restriction enzymes to cut it out, and then you use the polymerase chain reaction to replicate DNA. You then modify the gene to work in a more desirable way once inside the recipient organism. This step isn't always needed. And then you deliver the new gene, which is called the transgene, into the cells of the organism that's receiving it. And this is called transformation. So a genetically modified organism is an organism that is the result of genetic engineering. GMOs, genetically modified plants, usually are used in a variety of human activities to improve the quality of life or to improve productivity. GMOs can be microbes or plants or animals. Most often they would be plants. The importance of genetic engineering and GMOs, they affect many different aspects of our life and our environment. They play a huge role in the synthesis of medical drugs and in the production of new crops. They also play a role in cloning and in stem cell research. In medicine, two aspects of genetic engineering I'll discuss them. Firstly, the production of artificial hormones such as insulin using genetic engineering, and secondly, the production of vaccines. So to produce a hormone such as insulin, we can actually produce human insulin synthetically through genetic engineering using recombinant DNA technology. So recombinant DNA is DNA that has other or new pieces of DNA inserted in it to create a new altered piece of DNA. And we can do this because insulin is a hormone and hormones are proteins. So we can get the gene that codes for the production of insulin. This is how we do it. The DNA within the gene coding for the production of insulin is removed from a healthy human pancreatic cell. So a healthy cell that produces insulin. Enzymes called restriction enzymes then cut this DNA into segments to isolate the specific gene. Sorry, the word gene is missing there. Then a bacterium called E. coli, which is usually present normally in a human intestine, is used to produce insulin. So for this insulin process, they use lab-grown E. coli as opposed to E. coli actually found in our gut. The plasmids are removed from the E. coli bacteria. We know that plasmids are circular DNA that is unique from the genomic DNA, the nuclear DNA of bacteria. The restriction enzymes are also used to cut the plasmids. So we can see here if the plasmid is usually a circular shape, then cut by a restriction enzyme to be laid out into one straight line. The insulin gene is then added on the end of the plasmid, and then it is made back into a circular DNA and is now recombinant plasmid because this gene has been inserted. The human gene that's, as I said, is coding for the production of insulin is then inserted into the open bacterial plasmid, and this is controlled by the by the enzyme DNA ligase. The plasmid joins to form the ring shape again, and the human gene is now part of this new recombinant DNA. This recombinant DNA is then placed back into the host cell, the E. coli. It's obviously not placed into a human cell because human cells don't have plasmids. It's placed into the E. coli. The E. coli accepts the new gene as part of its genetic material because it's inside the plasmid, and the E. coli begins to produce insulin. The bacteria are placed in a spatial fermentation tank where they reproduce really quickly, and the replication of the desired gene takes place as soon as the E. coli reproduces. And because this E. coli is continually reproducing, it's continually producing insulin. Humans will then extract this insulin and take it as pills or hormones or whatever necessary. So this is the process where you have a human pancreatic cell, you take out the insulin producing gene. You have a bacterial cell, you take out the plasmid, cut the plasmid, insert the recombinant DNA. You then put it back into the bacterium and you now have a recombinant bacterium. This bacteria reproduces over and over, and it continues to produce human insulin. The human insulin is then extracted and purified, and that's how we get our insulin. So the two important enzymes for this is the restriction enzymes and DNA ligase. This is another diagram of the same process. Vaccines can also be produced using genetic engineering. So we can modify certain bacteria and plants to produce antigens for certain viruses. Antigens are just protein molecules that occur on the surface of cells, and they act as identifying markers. So in vaccines, they used to trigger an immune response to the body because the body recognizes the antigen. So the body sees, okay, this is a foreign body. It shouldn't be here. We need to get rid of this. And the body begins to produce antibodies, which will get rid of the virus or the harmful substance. The antibodies provide immunity from infection by that particular virus, because I think you might have learned by now that once your body has produced specific antibodies, they'll continually be there. It won't need to, if the virus or the harmful substance comes again, the body won't need to look, say, okay, what are these antigens? What antibodies do I need to produce? Let me produce the antibodies and then kill, which takes a long process. The antibodies are already there, and they already remember that, okay, I work for the specific virus. So the next time you get the virus, you can immediately get rid of it instead of undergoing the whole process, which means that it's a lot quicker to fight off. So the virus hasn't multiplied as much, which is why they provide immunity from the infection. So antigens of the hepatitis B virus are an example of antigens produced by the genetic engineering. So what would happen is the vaccine containing the antigen, probably not containing the whole virus, usually just a part of the virus, but with the antigens, are injected into a person's body, and the person then recognizes or then creates antibodies to fight against this antigen, and it gets rid of it. And then the next time the person gets the actual disease, as opposed to just a vaccine, they're already ready to fight it off rather than needing to undergo the whole process. That's why vaccines, that's why some people say, okay, vaccines are dangerous because the virus is actually being injected into the person's body. But we can see here that it's a less harmful form of the virus. So only a part of the virus, or a weakened virus, or a very small amount or something, which allows the body time to fight it off and allows the body immunity for the next time you get the virus. This is just another diagram showing it. You can also use gene therapy, which is a very important part of genetic engineering. Gene therapy is an experimental genetic engineering technique that replaces a faulty gene or adds a new gene in an attempt to cure disease or to improve the body's ability to fight disease. So the cell containing the faulty gene, either a blocking gene could be put in and the cell could be functioning normally, or the cell doesn't have a gene at all and a functioning gene will be put in and the cell then functions normally. Currently, the only way to cure a genetic disorder is at the embryonic stage, but gene therapy actually allows genetic disorders to be cured at any stage in a person's life. How are new genes introduced into the body? Well, we know that they're introduced usually through things such as vectors, which are usually viruses or bacteria. They deliver the genes to the cells of the body. Usually, the two ways of delivery are direct delivery and cell-based delivery. Direct delivery is that the vector is injected or given by an IV directly into a specific tissue in the body, where it's taken up by the individual cells of this tissue. It also can be through cell-based delivery, where the patient's cells are removed from the patient's body. Then they're exposed to the vector in the lab. Once the cells now contain the vector, they're then returned to the patient's body. And if the treatment is successful, the new gene delivered by the vector will make a functioning protein. You use the patient's actual cells to put in the vector rather than just a random amount of cells, a random number of cells, because if it was just random cells, not the actual patient's cells, there could be risk of rejection. So what viruses do to infect hosts is that they make their host cells make their protein for them. So they actually put their DNA into the host cell's DNA. So now researchers are using that technique, that ability of viruses, to be beneficial for humans, so that viruses will still put their DNA into the host cell, but their DNA has now been modified to have desirable traits or make desirable proteins. And geneticists will take out the bad parts of the virus, so the harmful parts, so that now the cells have the ability to make the good gene. These are the two different deliveries: direct delivery and cell-based delivery. We also can use genetic engineering to produce genetically modified crops. So genes for desirable traits are identified and they're isolated from a whole lot of different organisms. They're then artificially inserted into the plant cells, which will develop into the adult plant with the desirable traits. There are two ways of transferring the required genes to the plant cells or to the plant tissue. Firstly, through a gene gun, which shoots small metal pellets coated with the required DNA directly into the isolated plant cells. We spoke about this a little bit during the video on, I think it was on asexual reproduction in plants. The plant cells are clones because they're all genetically identical, and each cell develops into a new plant that displays these desired traits. Agrobacterium whatever, this bacterium can also be another way to genetically engineer plants. We can use this bacterium as a vector to transfer recombinant plasmids to the plant tissues. So the recombinant plasmids that we spoke about earlier during the insulin thing. The process is very similar. You guys can read through it. Genetic engineering and GMOs are really important for a number of different things. Firstly, polyploidy. This we spoke about a little bit during the meiosis video. It occurs due to abnormal meiosis where chromosomes don't separate properly during anaphase. Humans have deliberately manipulated chromosomes to form hybrids. This is only in plants. In humans, there are no polyploid humans. Polyploidy is basically, for those who don't know, is just the condition where instead of having 2 pairs of chromosomes per chromosome pair, there'll be like 4 sets of chromosomes, or 5 or 3 or whatever, just more than 2. So instead of being a diploid organism, it would be triploid or a quadruploid or tetraploid or so on. So humans have used this in durum wheat, for example, to produce tetraploid durum wheat, so 4N wheat instead of the normal 2N, and then I have 4 sets of chromosomes. Some of the advantages of being polyploidy is that the plants will have larger flowers, larger fruit, larger storage organs, fruit without seeds, or greater resistance against disease. So some of the advantages of just GMOs in general, it doesn't necessarily have to be polyploidy, is that the plants may have larger, better yields and stronger crops. This means that it's a lot cheaper for farmers to produce these plants with larger, better yields, because per every plant, there would be more fruit or more flowers or whatever it is. It's also cheaper for farmers in terms of producing crops that are genetically modified to be resistant to insects or pests, instead of needing to spray them with pesticides and insecticides. It's also a lot healthier because it reduces the spraying of pesticides and insecticides, and more environmentally friendly. Certain crops are genetically modified to be resistant to herbicides. Herbicides are weed killers, so then farmers can spray the weed killers freely without worrying about affecting the crops themselves. You can also produce improved crops such as higher yield, higher nutritional content, longer shelf life, larger size, a better texture, firmness, taste, more juice content, and plants can be seedless. Some GM crops can also survive in unfavorable conditions, so on land that is previously unproductive or infertile. Now GMOs can be planted there, which allows us to have a lot better efficient usage of our land. You can also use genetically modified sheep to produce a human in their milk that is used to treat certain produce a human protein in their milk, that is used to treat certain medical conditions. You can also use bacteria and other microorganisms to produce medicines, like we spoke about in the insulin thing, which is more ethically acceptable than to use lab animals or to research on embryos. Some of the disadvantages of GMOs, well, there are sometimes questions regarding the health of humans, how GMOs affect that health, and this is specifically relating to the production of substances by GMOs that can cause allergies in humans. It also decreases biodiversity because only certain genes will be introduced, and then these crops will be clones of each other, and they're continually using the land instead of using a whole variety of species or one species with a large gene pool. And also these crops are planted on places where indigenous species are removed, which again decreases biodiversity. the undeniable effect of the new gene and organisms in the environment where the GMO is cultivated. So the effects of a GMO might be quite unpredictable and uncontrolled, but a lot of people will argue this and will argue that, oh, we don't know what this gene is going to do, we don't know what effect it's going to have. But this is quite almost an uneducated opinion, because this gene isn't going to affect other organisms. It's only affected the organism in which it's been inserted into. Another disadvantage is that the cost of a modified seed can increase drastically and become really, really expensive, especially if it's only produced by a few companies, and farmers have to then purchase new seeds each year. So usually farmers would buy seeds once and then collect the seeds from the new plants that have grown. But now some genetically modified plants may actually be seedless, which means farmers have to buy them each year. Or some companies have monopolies over this genetically modified seed, which means that they require the farmers not to collect the seeds, but require them, they actually have to rebuy seeds each year, and they can make it really, really expensive. And it's also expensive to use this biotechnology to genetically modify crops, which obviously can't be used by people or by companies that can't afford it. Some ethical issues, we'll also look at the moral values, so whether it's right or wrong to actually mix genes of different organisms, different species, etc. But this is, again, quite a controversial issue. No one can really say with 100% certainty this is right or this is wrong. And people also question the right of humans to change the natural order of life, to like play God, is quite a common phrase. So then if we just look at biotechnology, this is a broad term describing a field of study that uses living organisms and their biological processes to develop technology and products aimed to improve the quality of human life. So cloning is the process in which a genetically identical replica of a molecule, like DNA, or a cell, or an entire organism is created. There are three types of cloning: DNA cloning, therapeutic cloning, and reproductive cloning. So DNA cloning is just the transfer of a segment of DNA from one organism to a self-replicating structure, such as a bacterial plasmid. So it's literally cloning of just simply DNA. Therapeutic cloning is also called embryo cloning, where human embryos are cloned and produced over and over for research purpose. And the aim is to harvest stem cells in order to study human development as well as to treat diseases. So this is basically used for research. Stem cells are basically the meristematic cells of humans, so the undifferentiated cells in humans that still can specialize into new cells, into any cells. The third type of cloning is reproductive cloning. This is the technique used to produce an actual animal with the same DNA as another animal. For example, I'm sure some of you know of Dolly the sheep. Embryo splitting is when a zygote, so a single cell, a diploid cell, divides by mitosis to form an embryo. At an early stage, the embryo is split into separate identical embryos, which are implanted into other females for further development until birth. So a zygote naturally divides by mitosis to form an embryo. This is not a human-controlled process. That'll always happen. The human part of this is when the embryo is split into separate identical embryos, so 2 or 3 or 4. And then each of these embryos will be implanted into other females for further development until birth. So the females may not be the ones who actually have their genetic information within the organism. It could be just a surrogate female. And then we go into nuclear transfer. This can be embryonic or non-embryonic. If it's embryonic, this is when cells are extracted from young embryos. The cells are treated to develop into separate embryos, and the embryos are implanted into the uterus. So a lot of identical offspring with the desired characteristics can be produced from cells extracted from the same embryo. So one cell cells would be extracted from one single embryo, and then many different new embryos will be produced. They'll all be genetically identical. You can also use non-embryonic nuclear transfer, which is also called somatic cell nuclear transfer. This involves the nucleus of a normal somatic body cell as opposed to the nucleus of a gamete, where combining nucleus of a gamete. And the first mammal clone from adult somatic cell was Dolly the sheep. So in reproductive cloning, the donor animal is the animal that actually provides the genetic material, and the surrogate animal is the animal that carries the fetus to birth. So they could be the same individual, but then it's usually just a normal pregnancy. If they're different animals, then this is often what we call cloning. So in the example of Dolly the sheep, Dolly was a white sheep, but they gave the black-faced sheep carried Dolly's embryo to birth. They just did that so that they can show that even though it's a black-faced sheep carrying the embryo, the sheep born is a white-faced sheep, which just shows skeptics and other people that the black-faced sheep actually didn't have any effect on the DNA of the offspring. The offspring was DNA was actually from the white-faced sheep itself. So what happens is a black-faced sheep donates its egg. The nucleus is removed from the egg, so the only thing left in the egg is the all the other organelles, but not the genetic material, except the mitochondrial genetic material, but that's really another issue. So now we've got this nucleus, this denucleated egg. We then take a cell from a white-faced sheep, so this would be the donor sheep. We then fuse this cell. It's a somatic cell, not a gamete cell, so it's already diploid. They fuse this entire cell with the egg of the black-faced sheep. So now this egg has been fused with the cell, and it now has the genetic information of the white-faced sheep. This egg fused within the cell now keeps on replicating and replicating to form an embryo. This embryo is then implanted into the black-faced sheep. It could be the same black-faced sheep, it could be a different black-faced sheep, but usually it's the same black-faced sheep, simply because they had to use the egg of a black-faced sheep and they had to use a surrogate black-faced sheep so that the egg would implant. If you had the black-faced sheep egg and then you wanted to implant it in a white-faced sheep, this wouldn't work. It would be like implanting a gorilla egg into a human, like it's different species. It doesn't work. So the egg has to be implanted into an animal of the same species. Once this egg that contains the genetic information of the white-faced sheep is implanted into the black-faced sheep, it grows until birth, and the black-faced sheep then gives birth to it. But the offspring born has the identical genetic information to this white-faced sheep because it came from a somatic cell of a white-faced sheep. So the black-faced sheep was the donor of the egg, and it was the surrogate, but the white-faced sheep was the donor of the genetic material, and that's the species of the baby that was born. So that's basically the process of cloning. They won't usually ask you to describe the entire process, but they may give you something like a picture like this and ask you to explain in your own words or answer some questions on it. This heading of cloning isn't really the correct heading, but basically what this picture is just saying is that you can use a donor egg, which has mitochondria, and then you can use the patient's nucleus, the ones who want to have the child, take their nucleus, so their genetic information, insert it into the donor egg, which has been denucleated or enucleated, so that this egg has the mitochondrial information of the donor, but the genetic information of the patient, and you can then fertilize it with the patient's sperm and implant it in the patient female or in the donor female, the surrogate female, it doesn't really matter. And what happens is at birth, the offspring has the genetic information of the parents. It's now not an exact clone because fertilization has occurred. It wasn't a somatic cell, but it has the genetic information of the patient couple, but it has the mitochondrial DNA of the donor couple. So basically, what this slide is saying is just this is another process of genetic engineering, and that the child actually has their DNA from its mother, its biological genetic mother, its biological father, and it has mitochondrial DNA from the donor. So it actually has DNA from 3 people. That's all this slide is saying. So that's basically the end of genetics. There was quite a lot of important information on this presentation, so have another look through it. And if you have any questions, you're welcome to ask me.